welcome to the next in our webinar series. This one is How Did Macromolecules Gain Function? And well, on my notes it was going to be Lauren and Irene, but we now have a cast of thousands who might present at any moment, which is wonderful. Just a couple of admin points to deal with. There is a link in the event for this webinar to the slides. However, because the science is moving forward so quickly, these slides are now out of date, but in the next 10 minutes or so, an even newer version of slides will be up there. So just give me a moment and we'll update that. Secondly, we had a question the other day from one of the participants asking where they could find the link to the previous recorded webinars. So just to clarify that, in the events section, if you click on more events and then go back through the list, each of the events at the bottom of it, when you click on them, should be a link to the YouTube version of that. And then finally, the fact that we have recordings of previous events should be your clue that this is a recorded event, and everything that you say and type will be visible on the internet. So with that, Lauren.
Hi, everybody. I'm Lauren Williams, and sitting on my left here is Nick Hudd. We're both from Georgia Tech, and then hopefully you can see Irene Chen, who is at UC Santa Barbara. And basically our task here is to help develop the NASA astrobiology roadmap. We're trying to work out a document, or part of a document that will help guide us and other researchers and NASA into thinking intelligently and funding intelligently science related to the origin of life. So our small task here is to think about how did macromolecules gain function. And what I've started here is just a picture of the ribosome, which is a functional assembly and also thought to be one of the oldest assemblies in biology. So in a really basic way, we could say, where did this come from? How did these large polymers, where did they come from, and how did they become functional? And I thought it might be good, since probably because I'm just a reductionist by nature, is to start thinking really carefully about what we mean by function. What does it mean to talk about functional molecules? But actually, before I get very far, I want to talk about tomorrow's webinar. There's a hopefully most of you know about this already, but there is a webinar tomorrow, which is very closely related. In fact, it's almost integrated with the one we have today, which is being conducted by Uli and Burkhardt, and it's going to be the same time, same place, and that one's how did peptide meet nucleotide, which is similar, if you think about it, to how did macromolecules gain function. It's closely related. So hopefully those of you here can also make it to that seminar, or that webinar too. Okay, so what I want to do though is talk a little bit about function. I mean, I'm going to talk about function, how I would like to think about function, and then actually Nick is going to talk about what he thinks about function. Then Irene is going to talk about a little bit about. So what we're going to really do is start by saying, what do we mean by function when we ask how macromolecules achieve function? And so this is the list of us, some of us who have been working on this document. I probably need to add more people to this. And of course, everyone's welcome. We want the more people we have contribute to this effort, the better it will be. So if you haven't actually actively started working with us on this, and you're interested, please join us. Okay, so I want to talk about function from something from my biochemistry class, trypsin. Because trypsin has function. I think we can all agree that trypsin has function. It catalyzes a chemical transformation. And so when we talk about trypsin having function, we say, number one, it folds into three-dimensional structure. So that's part of trypsin function. It binds to a specific substrate. It stabilizes transition states and intermediates, and it is regulated. So I think in modern biology, we can kind of probably agree we know what function is. It has to do either if we're talking about proteins, it has to do with catalysis and folding and that activity, or if we're talking about genes, it has to do with sequences that contain information. So I think it's pretty easy when we look at modern biology for us to think about function. But when we think about ancient biology, or even maybe before biology, abiotic process, what does function mean when we're talking about abiotic systems or early biotic systems? And so if we think about that in terms of trypsin, we can break that down to say, what is three-dimensional structure? And that's basically self-assembly. I mean, trypsin achieves its three-dimensional structure by self-assembly, and then it binds to specific substrates by assembly between protein and substrate. It stabilizes transition states and intermediates, and that has to do with assembly between the protein and transition states and intermediates. And then its regulation also has to do with assembly. So I think my thought is people are having a hard time logging on. I'm seeing that. Anyway, I guess somebody's working on that.
Can somebody log in right now? Get in.
So okay, I'll just proceed with this. So anyway, when I think about function on a really detailed molecular level, as a reductionist, I guess, I'm thinking about assembly molecules with complementary arrays of molecular interactions, as in DNA base pairing, or in protein beta sheet assembly. Those kind of really beautiful arrays of complementary functional groups that can interact. So in one sense, we can define biology as an extremely elaborate system of assembly and self-assembly. So when you hard-boil an egg, basically, you're just destroying all of that assembly. That's why you can never unboil the egg, and that's why it turns white. Basically, it all has to do with assembly. So now I want to just project that back and think about an abiotic system. So we could think about Titan, we could think about the ancient Earth, we could just some abiotic system. I think most of us here will agree that abiotic systems can generate organic molecules, small organic molecules. There's just a lot of evidence that can happen. And so this is my schematic of a collection of organic molecules. I only have two, but it could be more, it could be one. It's not really that important. We have some totally abiotic system that generates small molecules. And those small molecules can self-assemble, some of them. Or maybe it's not self-assembly, maybe it's assembly between molecules. But anyway, I think it's clear that organic molecules can be generated in abiotic systems, and some of those molecules are certainly capable of. And the interesting thing is that assembled molecules have different properties than unassembled molecules. We know that they're basically more stable to chemical degradation, enzymatic degradation, irradiation. Basically, once a molecule is assembled, it lives longer than a molecule that is not assembled. And so I would suggest that this is kind of the kickstart of evolution. You could have a system where you really don't have catalysis, you don't have polymers, you don't have Darwinian evolution, but you could easily have self-assembling molecules, and then the populations of molecules are going to be shifted depending on the assembly process. And so, to me, this is sort of, I would say, would be the kickstart of evolution, is that assembly can come very early and is not all that hard to get. And I think if and this is not just an esoteric, unanswerable question, we could look at Titan, for example, and my prediction would be that evolution has already kick-started on Titan. We know we have rich organic chemistry. We even have polymers on Titan, and ultimately, when we can find out a lot about Titan, NASA has to do the assays to get the instrumentation there. But the prediction would be that there are self-assembling molecules on Titan, and that the molecular populations are perturbed by that assembly. Basically, that there's an enrichment of assembly molecules on Titan. There's dunes of organic material on Titan, and these lakes are saturated with these hydrocarbon lakes are saturated, and we have photochemistry. So we have synthesis and degradation. We have all of that going on. So this would be a really, I think, an excellent sort of abiotic system to basically say is not to suggest maybe that we have Darwinian evolution going on there, but that the chemistry of Titan is in part sort of primed for biology because of this kind of amplification of self-assembling molecules. So that's a minimalist, and if anybody has any comments, that's a minimalist definition.
Is it possible to butt in here? I wasn't sure if...
Yeah, that's what we're after.
Okay, yeah, James Lyons, ASU. Just your Titan analogy. Mostly what you're doing on Titan is creating aerosols, particulates in the atmosphere, and those are settling onto the surface. So your chemistry is really going to be, I think, dictated a lot by those particle surfaces.
Well, yes, that's why I did write surface, in the lakes on the surface, or in the atmosphere of Titan. So, yeah, I mean, my understanding I was just out at the Icy Worlds group at JPL, and they were, looking at saturated solutions of hydrocarbons mimicking these lakes, and at least the information I had from them that there were probably high concentrations of organics of some kind. Exactly, I don't think in the lakes. That was sort of my basis for saying that.
Okay.
But we can talk about that. All right. So anyway, that's kind of a minimal.
You want to see if there's any questions for you?
Yeah, are there questions on that? Because we're going to now change, and Nick is going to talk a little bit about sort of a higher level definition of assembly. Okay. I'm saying no.
No. Okay, so Lauren asked, how would I address this question about this emergence of function? And I've been thinking about this a while, and with Sarah Walker, who's a postdoc with us for a year, you might know her, we worked on a simulation, and I'm just going to go through just a few slides to show you what we're thinking, but it's more of a backdrop of what I think are some of the big questions here. And I start off with this slide, which is showing that we're expecting that we have polymers and monomers, and that these go together to either make more polymers or somehow they're templating to replicate themselves. And I think that this captures the type of system that Lauren is talking about. When he talks about functional systems, Lauren's saying the minimal functional system you have is that you have to have recognition, you have to have assembly, you have to have folding. And I agree with him about that. But what I also countered to him was that I think that when a lot of people think about function, they think about the emergence of sequences that can do something, right, beyond just what the polymer, intrinsically does, like, form a stable structure. And so I think when we think of function, we have to come up with models for how do we, say, generate molecules or how do we favor molecules that have sequences that can catalyze reaction. And so what we did was we looked at a model where we have polymers down on a surface in a 2D, and they diffuse around, and then we have monomers that can move around too, and they're polymerizing. I'll go to the next slide. And just show you some of the things that we see come out of these that I think give us an idea about what is possible and also what we're going for. If you have a system of monomers that can spontaneously polymerize and replicate, then what you're going to do is very quickly you run out of resources and you reach a steady state level of the number of polymers that can be supported by the system. And then it becomes limiting in the number of monomers that you have. And if you consider that there could be lots of sequences out there, but none of them are functional to start with, then nobody has a selective advantage. And sequences stochastically can come and die off and live on, and some will build up. And if you look at this down here on the two-dimensional grid here, because we have limited diffusion, what happens is that you get colonies of polymers that are building up and over a number of cycles. Now, there's no functionality in these beyond what Lauren had said. Lauren had said that monomers that can recognize each other come together. So that is implied in this, that we have that level of functionality. But what then we did was we said, what if a sequence comes along, and we call this an azyme, and the idea is that there are monomers that are A and B that pair together. And the idea is that if you have this polymer that can generate a monomer that's in short supply, we said, in a system like this, does that polymer have a selective advantage? And the answer is absolutely yes. Over a wide range of parameters, which I won't go into, but these have to do with the lifetime of these polymers in the system and the rate at which the monomers move around, diffuse, and that. What we're seeing is that a polymer that has a function has a much longer average lifetime than just random sequences in the mix. Even though these aren't compartmentalized, there's just limited diffusion. And the population, the number of polymers that are steady state, that have a function versus those that don't, is also much higher on average. And so, just to look at this in a spatial map, what you see is that what we have here is starting off, we have on the green line here is this: if you have no functional sequence, you go up and you reach a steady state level, and then forever you have the same number of polymers. If at this point right here along comes a functional polymer that can generate a monomer in short supply, then the whole population overall benefits from that until the other monomer is in short supply. And then you can do a scenario where a sequence comes along where the other monomer that's in short supply is catalyzed by some functional polymer, and then everybody builds up in their populations. So the reason that I bring this up is that I think that what we need to address in looking at functionality emerging in polymers is a very wide range of functions have to get their foothold in order for life to really come about. I mean, what Lauren's talking about is fine with respect to what are the basic building blocks that you need. You need building blocks that can recognize each other, pack together well, that can form a backbone, that can fold and that. And so right away we think peptides fit the bill, nucleic acids fit the bill, that's great. But then we have to select sequence from there, and we have to select a lot of sequences. What I think is that we have to have life before we even get to, say, cellular life. It depends on your model of when you want compartmentalization. But before you have something at the level, say, of the ribosome, I think still there would have been a lot of enzymes that came first that were selected out of maybe non-templated polymers, right? Ones that folded and were stable but still were doing something that was good for them or their environment. And so, what I see is a need is to, especially experimentally, demonstrate that this is the case. I think, there's a lot of focus that's been on getting a replicator and that's good, but we need to be thinking about the systems that would allow many different functions to take hold in the soup. And this is, when I think about this, it connects from the top-down work of Carl Woese. And what Woese says is that, when he looks back at the evolution of the cell based upon his genetic analysis, he comes to the conclusion that there must have been many cooperating polymers because there were so many problems that needed to be solved. And so, in my mind, when we're looking at function coming out of these polymers, we have to, in the earliest stages of life, we have to look for a system that would allow, right, the coexistence, the non-selfish, say, cooperation of many functional polymers. So that's my stick on that.
Okay. And then, if people want to weigh in, and Irene has some ideas on this also, I believe.
Okay. Yeah. Does anybody want to say anything or comment? I know.
Well, I just one quick question, Nick. Were those the azyme, bzyme? Were those actual experimental results? What were you showing there?
No. These are simulations. These are, the idea that
computer simulation. Okay.
Yeah, it's all computer simulations, and that's why I did emphasize. Yeah, I think what we really got to go for is a push for experimental systems that can demonstrate emergence and coexistence of multiple functions. I think that's important.
Absolutely.
Nick, do you want to pick up? I think that David had made points about assembly being downhill and the thermodynamics.
Oh yeah. Yeah, so David Ross wrote in there, should we not be considering the thermodynamics of assembly? Oh, absolutely. That's something Lauren and I are both big fans of. How do you get those polymers to start with? And I think that having a thermodynamically favored assembly is superior to one that you have to rely upon kinetics of activation, polymerization. Because in the beginning it's got to be really simple. And so if you have something that's thermodynamically favored, then you can have the system run for a long time, and then the formation of your polymers would be more akin to, say, crystallization, right, rather than radical polymerization. And I think that that's important. And what's also really important about thermodynamics of assembly that goes along with what Lauren's talking about, in making stable structures, you want to find the energetically favored structure, and you'd like that to be something that, say, is folded and has lots of interactions. And so then having, say, backbone chemistries that are thermodynamically favored, at least in some parts of a cycle, is to me the most direct way that you will get to a solution to this problem before you have all the complex machinery that we know now is in the cell.
Yeah, sort of on a big question, I would say, is, in fact, in this document is possibly one of the issues we could treat is the issue of thermodynamics versus kinetic traps. Modern biopolymers like DNA and RNA are high energy kinetic traps, and is that a good model for ancient systems? Should they be closer to equilibrium or not? So there's another question here. Bruce Springsteen's,
Greg Springsteen.
Sorry. Are you going to sing for us? Okay. Greg says there's a separate document called Synthesis and Assembly of Populations of Stable Oligomers that he presented last week. So he's looking for input on that document too. Yeah, let me say that there's a series of documents that are kind of closely related and overlapping, and all of this will ultimately be funneled into this document called the Roadmap. And so we're just working on that process here to get some parts of that document together. Okay, so Irene, did you have some comments?
Yeah, I'll just make one quick comment, and then I also want to say, reading the chat window, it looks like people do want you to go over the document itself, so that might be a good idea. I'll leave a lot of time for you. So my one point is about this whole prelude about what is function and defining function. I think Nick started to get into this. I want to make sure that we have a pretty inclusive or broad perspective on what function is. It could be the chemical perspective of assembly and catalysis, but also even really simple physical phenomena like structural. We think of structural proteins, and maybe if we have structural polymers also, that could be helpful in some way. So just anything that enhances the fitness of an organism an organism, I guess, broadly defined. So on the physical side, even osmotic effects that we've looked at previously could be of interest in terms of function. And then on the biological side, there's things like gene regulation, where you're just talking about response to chemicals or even buffering and things like that. Those simpler functions might actually be easier to achieve and still interesting for life. That's all. That's the only point I wanted to make. So I'll give it back to you, Lauren.
Yeah.
Okay.
Hi. This is.
So go ahead. Somebody else comment?
So this is Andy Ellington. Hi. So I sort of came in a little bit late, but I agree. Maybe it would be good to go over the document because I went and clicked on the document window, and it's a rather general document that deals with many important issues. And there is this, perhaps slight impression that if we deal with individuals' research in a seminar that's essentially about discussing the document, that what we're really doing is trying to write individuals' research into the document, which would probably not be a good thing.
We don't want to do that.
We don't want to do that.
No.
Well, you could have fooled me, dudes. I mean, come on.
No, we're just trying to get some conversation going. So
what we have actually,
this is one of our perspectives on it, so let's bring it up.
What we have, actually, the next slide. Okay, now I have taken the document here, and I have, so the next slides are actually, what I did is I took the document and I pasted it into this PowerPoint presentation. And what I did is, so that's what basically these next slides, which I'm flipping forward through and reverse through, are the actual document. So what we were trying to do is sort of generate a kind of discussion and some sort of framework for discussing the document. So, and some of this, actually, and also I sent the document out to a list of people who I just made up a list early in the week and sent it out to them, and I got comments by some people, and so those are some of these initials here. This is Uli, and this is me. So I got some things back, and so if I didn't send it to you, then I'm sorry. Please send me email, and in the next iterations, I'll get you on that email. Andy, I think I did send it to you. So
I'm sure people send me stuff all the time that I don't read. So that's not it's not your fault if I don't read and comment. But I still just think that we should try and keep this as neutral as possible as we move forward.
Yeah, I have to say that. Okay, all right, let's do it. So okay, so maybe these are comments that we had. So this is basically then. Sorry, having some. We can go through
everybody. I just wanted to make a quick note. This is Lindsay Hayes. I just wanted to say that for any and all of these roadmap papers, we appreciate contributions from everybody. So, particularly, Andy, if you feel like this is too centered on one particular type of research or one particular group's type of research, we really, really are encouraging people to make comments and get involved in this whole process to, make sure that their particular interests are included in each of these white papers, and therefore will have a place in the final document that comes out of this.
Mm-hmm. And Lindsay, let me just chip in one other thought, because I think we have several people here who haven't been to previous webinars, so I just wanted to explain some terribly mechanical things. On the front of the website, you'll find a link to each of the white papers or concept papers that have been presented, and the one that Lauren and Co are presenting now is at the top of the list. Prior to the webinar, it's in read-only mode. And the only reason I'm saying this is you don't actually have to email it to anyone. Everyone has access to it. As soon as the webinar finishes, we flip the switch and it goes into comment mode. And all you need is a Google Docs account. So everyone can read it right now and should be able to comment on it in half an hour's time. Okay?
Yeah. So Andy, if I could just ask real quick. So let's assume that just besides being lazy, there are some of us who have not had a chance to really, chime in until end of term. When is the comment portion closed for all the documents, and at what point do you guys start taking action?
Actually, can I field that question, Andy?
Yeah, sure.
that question from Andy to Andy. So the current schedule that we have, you can actually, if you go back and look on the same page that the Astrobiology Futures page, Michael New's presentation, the way forward, sort of gives you a sense of the timing. The goal is we're going to finish up with our final presentations sometime in mid-January. We're going to leave them open for a bit longer for comments and such, but then the goal is in about starting in February and through sort of mid-March to start doing edits of these and to really start bringing them together, either in some in-person or some virtual way. We haven't quite outlined that yet, but the idea is basically it's at some point we're going to bring it together, and that's probably going to be, like you said, like you're asking for a date, sometime early February through early March.
Okay, so if we want to get our chits in, we need to get them in by mid-January, early February, something like that?
Yep.
Okay.
Thank you.
Back to our presenters.
Okay. So what the note here, and this was actually not part of the document. This was a note I wrote into the document that I sent out to everybody on Monday, I think. But what I would like, what I propose to do is ask if we could try to, and maybe people don't agree with this, but this was just an idea I had, was to try to establish a sort of a temporal order to increasing complexity of function. And I can see now, actually, from listening to Irene and other people, that actually the list of function I have here is not complete, but so that basically we could write sort of our broad definition of function, and then we could maybe try to see if we could put things in a temporal order. That was just an idea I had, that if people want to weigh in on it, they can.
I think that's an excellent idea.
Okay. I think that's a terrible idea.
Okay.
Well, okay, so what should we do instead?
I don't know, but I mean, do we need temporal ordering? Can't we just say, hey, we're going to require the evolution of functionality. The evolution of functionality may have occurred by a variety of different pathways. It doesn't have to occur by this pathway. The proof's in the pudding. How do you get to the functional biopolymer or the functional early replicator or whatever you want? And so there may be elements of it, and these are some of those elements, but other, depending upon the system, depending upon the polymer, depending upon, what mechanism or scenario you pick, that might have occurred in different orders.
So maybe the right way to think about it is not a temporal order, but a probability, relative probabilities, or ease of emergence.
Which again, I think would be system specific. Yeah.
Okay, but then there's levels of it, and that's what Lauren and I were trying to, when we first started talking about it, we were talking on very different levels. Lauren was talking about function that was more of, associations, even monomer associations, and I was thinking that when people think about emergent of function, they're thinking about, polymers with sequences that have activity, right? But those are, one of those is, say, on the nature of the molecule themselves, and the other is kind of information part of a molecule, in different spaces.
But again, those both assume I would argue those both assume particular mechanisms. I mean, we both know, from the literature and from experiment, there are a wide variety of mechanisms by which people assume functionality arises, some of which pass through biopolymers, some of which don't, and some of which can pass through other mechanisms and then become biopolymers. And so, since in the end we're, I think, all interested in what either was likely to happen or could happen elsewhere, I would say unconstraining this problem in terms of temporal order is necessary. It's going to be system specific, and it's probably up to investigators to specify their system or specify their mechanism, and then apply these different sort of functionalities as they wish.
I don't see why we're getting so hung up on temporal order. I mean, it's increasing complexity that matters, and you could just drop the temporal order, and it's not going to change anything.
Exactly.
Okay, I would argue that even complexity is a loaded word, and that right,
temporal
and so
really loaded in this context.
Right. So temporal is really loaded. Complexity is still loaded, and one can imagine specific binding prior to self-assembly, or specific binding being more probable than self-assembly, and so on and so on. Again, I think it's up to the characteristics of individual systems, and therefore up to individual investigators with individual systems to show how these play out. I'm uncomfortable with making general sweeping statements that have no, grounding in chemistry or molecules or whatnot that I could at least see my way fit to saying, oh yeah, that it happened that way and not that way.
I think it's just a framework. I don't think it's...
You have to say something. So Andy, give us a statement that you would agree with.
These are all interesting functionalities. It would be, can you individually or collectively show how to instantiate these functionalities with biopolymer systems of any sort? I mean, that would be a challenge to the community that doesn't require any grounding in temporal order or probability or complexity. It's just, we recognize these, they're important. How do you get there? There could be multiple pathways, and those pathways could be traversed differently by different molecular sets. There could be different probabilities depending upon the different molecular sets. I think it's an interesting list.
Mm-hmm. Okay, do you have more on the document? Do you want to go through?
Yeah, one of the things I have actually that I think got pushed off the page because actually Uli thought it was so obvious that I shouldn't even say it, was that all of this should be linked to experimentally addressable systems, so that whatever we're doing here needs to be linked to experiments. So that was another question. Okay, so now this is right out of the document. This is, we had the meeting at Wallops Island, and we, as a group, and I mean there were people online working on this, we came up with some that we thought were, or some of these look like statements that we thought were important in this document. And I haven't touched this, I just copied and pasted this directly into the thing. And I guess maybe why don't I just let people read this for a second before we go on.
Lauren, while people are reading that, maybe I could just make the comment that the point on on the previous slide, maybe it could be phrased as a hypothesis, and it could be like addressing the question of whether there are general general features to the temporal order or probabilities or whatever, general to multiple chemical systems. Maybe it could be phrased as a question to be investigated.
Yeah, that's a good idea. Are there, if people have gone through this, are there particular points in this list that people would like to discuss? Is there something here that people think should be stressed or should be struck?
These all seem like excellent questions to me because they're pretty open-ended, or even when they're asking for this or that, it's a pretty general this or that. So these seem like, to me, like really good questions to address to the community.
Okay. Anyone else? Comments? So I think when we look at these questions, though, we need to think about them in terms of what the real roadmap is going to be, because we cannot, I don't think, include a lot of this detail in the roadmap. I think the document we're looking at is probably going to be distilled down to maybe a couple sentences, probably. But we need to, this is a framework for us to think about this, but somehow this has to be tuned a little bit in conjunction with some of the other documents.
So I just wanted to also add here, this is Lindsay again, one of the things that we were really sort of trying to encourage with some of the, one of the earlier papers included a section that we called Things to Work On in the Coming 10 Years. And that kind of using, turning these great sub-questions into, where, what specifically do we think we want to work on, what specifically do we think we can get to, that kind of thing in the next 10 years might be something that you want to, a lens that you want to sort of view these questions through, and think about how you want to arrange your paper that way.
So Lindsay, that was sort of what I was a little worried about earlier. I know we're getting input from the community, but what I keep thinking is, people who haven't participated in these calls or worked on these documents are still out there. They'll eventually be invited, hopefully, to participate in the roadmap. And I keep worrying about the ideas we haven't seen or won't see as a result of these discussions. And so if we target specific areas of research or specific sort of projects even, rather than these very interesting open questions or even the previous slide of very interesting functionalities, that we're going to unintentionally include or unintentionally exclude people from the greater whole.
And we agree. This is actually something that Michael and Mary and I and Sean and Andy have been talking a lot about, is trying to make sure that not just questions or sub-questions or topics that could be, that could be into any of the individual papers that might be excluded, but even broader topics that aren't, that people feel might be missed. If you feel, that's part of the reason during the kickoff we showed the spider diagram that we did of the topic papers that we had, and we said, these are the topic papers that we have so far. If anybody feels like they can't see their research in here, or that they think that something was missed, we have encouraged and are continuing to encourage anybody to say, hey, I'm going to write a white paper on this. Do you think that'll fit in, that kind of thing? Bring some friends, some other people you work with to help you, get a topic paper written. Since we were going to be using these topic papers to assemble the thing, this is why we've been trying to get as much input from the community as possible. If you see something that's missing, please let us know. Try and work on a white paper or bring some people together or whatever.
Is that on the same time frame? So you would have to have that by mid-January, or do you need that sooner?
No, that's on the same time frame. We would hope that if you could get it, these are white papers, right? So they're relatively short, and we don't need any additional research really going into them. But the hope is that if you guys could get a white paper together, if you guys feel like there's some topic, we do still have some time at the end of January that is still available for white papers to be presented. So we're not. We would rather keep this going a little bit longer and make sure we get the right amount of feedback than cut it off and miss something.
Right. Okay. Well, that's great.
Yeah. And Andy, I see, just put up the link to the spider diagram. It's, again, if you go to that website, Astrobiology Future, you can see everything that's been done. And please, if you think that something's missing, we would really, really like to get that in.
Right. So I think that's a. Sorry, I think that's a great. I think that's a fine response, Lindsay. But it sort of a little bit misses my point, which is, for the people who don't participate, yeah, sure, I can go and try and cover the universe of ideas not represented. I'm not going to do a very good job of that. I keep thinking about, a younger Nick Hudd who, has these amazing, really novel ideas that probably nobody but Nick Hudd is thinking about. And if we weren't lucky enough to, capture the younger Nick Hudd early on, and they just happened across this call, how would we know that there's something there for them? I think in some ways the more general we are in stating the ideas, stating the problems, stating the questions, the more likely that we'll cast a broad net to find novel solutions, not just solutions by the community that already exists and is already participating, but by lots of other people. So that's why I get wary when it's like, well, let's talk about this aspect of research or that aspect of research or this particular order or whatnot. I really don't want to lose a broader community where there are fresh ideas that we may not have thought of.
So basically what you're trying to say is you want to be the voice of keeping the final roadmap, which we're actually calling the strategy, keeping that as broad as possible.
Yes.
Okay. We will definitely, that's a good point. And when we get to the stage of putting this all together, we will definitely try and do that. But in the meantime, part of the reason that we wanted to, get as many of the community involved is the hope that, somebody may know that other, the young Nick Hudd, as you put it, and get, and speak up for them, or, add a little, hey, maybe we should think about this or that kind of thing. But I see what you're saying. I think you've got a good point, and I understand what you're getting at, I think. So if does that sound good? Does that?
Yeah, sure. No, that's great.
Okay.
I'm just trying not to interrupt. Sorry.
Yeah, so that was, I guess if that's your point, I agree that keeping the final paper general is a good point, and we will try and do that.
Lauren, we can hear you, so go right ahead.
Sorry. I just want to say one thing. I mean, I guess maybe this was misunderstood, but in the beginning of this talk, I thought I should just try to present some things to stimulate the discussion. Honestly, that is not my research. That's not the research I do. It's just that the title of the talk was, the title of our paper was on assembly, and I thought, if we had sort of a detailed talk about what assembly was, it was on function. I just wanted to sort of direct the talk to the definition of function. But honestly, I didn't mean to give the impression that we were focusing it on some detailed kind of research. So if that was the impression that was given, then I'm sorry for that. Also, David Ross had a statement there about thermodynamics. And, what we should do, David, is just, during the editing process, just make sure your comments, I think your ideas are really good, and we should just make sure they get worked in. At this point, I don't think we can really edit it and add it in here, but that should definitely be here. So you just have to kind of keep after it and make sure that your comments get in here. Okay. Are there other comments and questions? Okay. I'm going to go to the next page.
Well,
okay.
Can you hear me?
Yes. Yeah. Sorry.
Hello. This is Lucas. I was curious about this question of what constitutes a function, and I think, while I don't think there's a strict, rigorous answer for that, but it might be useful in a document somewhere to have a short list of particular functions, like the one you had in your comment. I agree. I'm suspicious of putting it in any particular order, but I do think that for me, I'm still unclear exactly what constitutes what I suppose is a biological function as opposed to a chemical activity.
Right. I agree, and I think, I think it's something that this group could hash out. I actually, even though I was thinking in such a reductionist term, I actually like Irene's definition better than mine. Basically, a function is something that confers advantage. Is that what you said? How did you say that, Irene?
Something that increases the fitness of the organism.
Increases fitness. Yeah. So, and that could be, you could sort of use that definition on many, many levels. So I think that's a good but that's what this group needs to kind of hash out, because we can't, in this document, in the final document, we're not going to have a page to talk about what is function and what's not function. We have to be really concise and very general, and so that people who come along with things we haven't anticipated don't get ruled out because of it. Like Andy said, that's a really important consideration. It has to be general. It has to last for through a period of time where things are happening that we're not foreseeing.
Yeah. So Lauren, if I might say, I wasn't accusing you of presenting a specific view that was self-serving, but when I've done this before in other contexts, it's a little weird in this context because, the goal of the roadmap is more tenuous than, let's say, when we do it for DARPA and, the goal is, make a better bomb or whatever. So although the goals are more tenuous, I like the idea that if we have something we're attempting to achieve, and in this case, let's just call it a living system, and there may be a huge variety of pathways by which one could reach that living system, and also a huge number of questions, one of which we just heard brought up about chemical versus biological functionality and what that even means, that we leave it to the individual investigators to almost define their own terms, define their own paths, define a lot of things, rather than having sort of a groupthink, and even as genteel as this groupthink is, be what directs people to a particular path. And so that's the only thing I wanted to emphasize is I like the idea that this is essentially a giant biotechnology project geared towards making a living system. That's just my own view of it. You can have yours, but if that resonates with anybody, then sure, whatever gets us there is great.
Okay. Yeah, I think we're all on agreement with that. One thing I think that whatever the final document is, I think these discussions are very good because it, it helps us. I have to say that I never really thought about what is function carefully, I guess, until this morning, and so I think this process is good for all of us. Okay, I'm going to the next page. Are we all good on that? Okay. All right. So this is also out of the right out of the document, environment versus function, and I have to say I didn't really understand all of this. I just took it as it was. So let's just take a minute and read over this. And here, I confess, I put iron in there because that's my own work. I'll take that out. I was just like, I'm so interested in iron. I'll get that out. I'm sorry.
Iron's cool.
So this
Actually,
is where
if David's on the call, so David, what do you think about the question, what mathematical rules lead from equilibrium to non-equilibrium catalysis? Does that begin to sort of deal with your questions regarding equilibrium and kinetics of assembly?
Can you hear me?
Yeah.
Yeah.
Wasn't sure I was doing this properly. It gets there, perhaps. It's still a rather, that statement seems to me a little, it's just one statement, one sentence.
The other thing I was going to point to that exact statement too, and just say what you had said about thermodynamics and kinetics. Can you replace mathematical rules with, say, chemical rules, and then having in parentheses, thermodynamics, kinetics lead from equilibrium to non-equilibrium catalysis, or to function, right? But this is one thing there with, in the context of what Andy's been saying, I really agree with him that we don't want to be restricted because, it's an understatement to say that none of us, and even as a group, that we have all the answers and know what's coming in the next few years, right? That's for darn sure. But yet you still want to give a document where you do have, maybe some useful things in there to help direct in some way, such as don't violate the second law of thermodynamics, right?
That's a good start.
And I think that when you say what David wrote about thermodynamics and kinetics, I really like things like that because what you're saying is, we want to understand how you go to function, but ultimately these are chemicals, these are molecules, right? So they have to obey these rules of chemistry. Right? And
that's right.
This is also where when you're talking about, it's got to be connected to experiment. Okay, yes, but you could have a theoretician that is putting in, the kinetics and the thermodynamics and then defines these are the parameters in chemical space that you should be looking for. And that can be very useful. But I think
that's right.
You want to guide people away from, being free of any of that, where it's just A goes to B goes to C, and look, I've made a hypercycle. Right? You have to.
Yeah. They don't work, by the way, but if one looks at
they look good on paper, right? So we got to get the chemistry.
That's right. The foundation of all of this, it seems to me, are the thermodynamics and ultimately the kinetics. There are kinetic limitations. There are diffusion limitations. And those issues are, in my view, once again, the foundation of all of this. And when you have aqueous media, you got, that presents a number of problems that I think have to be dealt with directly with people who understand this sort of stuff.
Yeah, but I think that
any of this. I have no dog in this fight. This is just I want the science right. That's my view here.
But I think that's a great point. If only because A, I think hypercycles can work, and B, I think that there may be very restricted conditions under which they work, and the intersection between, where hypocycles work and what conditions they work under, which may very well be non-aqueous, non-diffusion, or slow diffusion conditions, is an interesting basic question. Could be approached mathematically and theoretically. Should be approached with experimental systems. But I think it's precisely the prejudices that you suggest, David, which are great prejudices, that should guide us but not limit us. They should be the basis for questions, which is how you've been posing them. But that's why I've sort of been pushing back just a little at a couple of points. It's like, wow, if anything, Lauren's diagrams are too seductive. If we get away from these seductive diagrams, what else is out there, and how can we state these more hypothetical or abstract questions so that we can get Lauren's diagrams and others as well, and then get to, as Nick was saying, these neat questions about, well, even if there was an experimental system or something where we could imagine instantiation, what subset or what universe would that define versus some other universe?
Yeah,
I guess I would like to give a slightly different perspective on this. I don't really see that the mathematical rules are going to be all that useful. I mean, I understand the fundamental basis of it, but what we really need is to address number 2 there, what functionalities are plausible in realistic geological environments, which by definition are aqueous environments, or subaerial, surface environments, things of that sort. But I mean, to me, this is. I would love to see this document really emphasize the experimentation and the connection to the geological environments. That's where I see real advancement here.
Well, I'll get off in a moment. I just want to say that obviously the laws of physics and chemistry ultimately are the basis of all of this, and so some
right, but they don't get violated in an experiment.
Oh no,
they get violated by theoreticians, of course. But.
Right, but you don't want to throw away all theoreticians, because if a theoretician will keep themselves constrained to the physical world and parameters and rules of chemistry, they might do something, good and show us the way as experimentalists, right? That's where
I tell you this. I am primarily a theoretician, so I'm just telling you this. Not trying to exclude myself from this, but I mean, what's really been lacking in this field is a connection to the geology, making the connection between what organic chemists think about in the lab, which is rarely to use an aqueous medium, to what you can actually do in an aqueous geological environment. I think that's something that's been sorely lacking in this area, especially when you look at the development of function as Lauren's initial list, that sort of progression of complexity. I don't see anybody doing that, and I think it's being mentioned here, and I think it should be emphasized.
The notion of a seductive figure is something that it's kind
Lauren
of
makes seductive figures. That's true.
Yeah.
Thank you.
Can I just interject at this point and say we're down to our last 2 or 3 minutes.
Okay,
are there any key points that you want to cover in that remaining time?
I would like to at least, we have one more slide. Let's at least try to get to it. That would be a good thing to do. So this is the next slide, and maybe just take, 60 seconds to read over this. Okay, so I'm going to assume, so is there anything here that somebody really wants, has a burning opinion about? Okay, let me just say that it was when I read this page that I thought, wow, we really got to think about what does function mean. And I think actually that this session has been really good at defining that. I'm gonna listen to this again and sort of write down some notes that we can put in. I think we can come up with some kind of definition of function. Whether that goes in the final document or not, I don't know, but at least it would make me feel good to try to write something down. And then I guess the other thing is that I want to stress to everybody is that I think NASA does a really good job at trying to get everybody involved, and this process is really open, and hopefully all of us can work on this and because, I mean, our community here needs to be well represented in the roadmap, and we need to do a good job on this process in order to have that work right for us.
Okay, thank you.
Can I actually, Andy, just wanted to say one quick thing. What I was going to say is there seems to be a lot of great discussion that has gone on today, and it seems like there are a lot of people who have some great ideas to put into this. So, Lauren, it's up to you, Lauren, Irene, other authors of this paper. If you guys, I mean, some of the other groups have had independent Google Hangouts or conference calls where they have actually, you know, you don't have to do this all through the commenting on the web or on the Google Docs if you want. If you feel like you would benefit from, you know, doing some of this offline, having a discussion, you know, that you guys want to do however and whenever you want, and then bringing it back together and putting it into the white paper, that's fine too. However you guys feel works best for you guys, go for it. We want to get all these great ideas incorporated, however that works. So thanks, Andy.
Okay. Can we get a list of all the email addresses of everybody who has been online here? I have most of them I'm seeing, but I'm not sure I have all of them.
Maybe the best way for that to be, actually, I don't know, you'll have to ask Mike, the host, if there's a way to do that. But one of the best ways you might do would be, do you want me to type your email address, or I don't know if you have that way, to type this into the chat, and that way anybody who wants to be a part of this group can just email you directly?
Idea.
I was also thinking that anybody that's on right now, they can type their emails into the chat, right?
Yeah.
Right.
Yep, that too.
So if everybody just did that right now, then Lauren would have that.
So this is, I'm going to do that. Yeah. Okay, so there's mine. Did I get it right? Yes. Okay.
Yeah.
All right.
Yeah, that's great. Then they're all showing up.
Yeah, do that. That's great.
It's got a record on there.
Good.
Okay, so just by way of wrapping up, and because I know there are several people here who haven't been through the process of commenting on the document, so a couple of thoughts. If any of the authors or anyone else involved in this would like to organize a Google Hangout or some other kind of meeting and they need help, just email us, support@NoInnovation, if you need any help on that, and we'll gladly help to organize that. The document itself is now open for commenting. What we ask you to do is you go to the document. To add a comment, you simply highlight the thing you want to comment on and right-click, and you'll find you can add a comment. It is really helpful, though, if you can log into your Google account first, just so it's got your name on it, because it makes it easier for people to get back to you, etc. So by all means, we can help you organize other meetings. Documents open for commenting. Everyone who wanted to looks like they have typed in their email address. Fantastic. Thank you very much.
There's another one tomorrow, same time. Please, I think it's basically going to in some ways be a continuation of this session. Maybe. I guess so. I'm not really sure, but I think it might be.
All right. Until tomorrow. Cheerio, everyone. Bye now.
Cheers.